Hi, thank you for taking a look at my presentation. My name is Sam Bratzman. I'm a master's student at BYU, and with the help of BYU and the Department of Environmental Quality, I've been studying algal blooms on Utah Lake for about the past year and a half. We're worried about these algal blooms because they can be really damaging to the lake ecosystem itself. There can be harmful effects on the local economy and for wildlife and people that come in direct contact with these blooms. There can be some pretty nasty toxins that are produced that can have really harmful effects on them. This has been a problem in Utah Lake for quite a while. And with the huge amount of growth that's projected for Utah Valley, there's a lot of potential for these blooms to happen more often and for them to be more intense when they do occur. So in our research, we had a lot of different questions. One of the main questions that we wanted to answer, though, is at what concentration or at what thresholds algal blooms are reduced. So basically, at what concentrations of nitrogen and phosphorus, we're going to see a lot less algal growth and therefore less of these really harmful algal blooms. So to answer this question, we performed experiments called bioassays and dilution bioassay. And basically what we did for this is we collected water from different sites around Utah Lake. We put them in these bottles that we call cubitaners, and we added different amounts of nitrogen and phosphorus to them. Once we added these nutrients, we allowed them to incubate for a couple of days so we can see how these samples respond to increased levels of nitrogen or phosphorus. So in doing this, we can see which nutrients are limiting growth and really just what's happening in a deeper level inside the lake. We collected a lot of data from these experiments, so I'm just going to share a small subset of the data we collected. The first thing that I'm going to share is from our dilution bioassay, which is a little bit different. Instead of just collecting the water samples and putting them into the cubitaners, we diluted them in a solution with a similar ionic concentration. In doing this, we get a better idea of where these thresholds are. So in looking at these two graphs, we're looking at chlorophyll A and phycocyanin. Chlorophyll A is a proxy measurement to measure how much cyanobacteria is in our samples. And cyanobacteria are pretty nasty because they are the ones that produce these toxins that we're so worried about. And also phycocyanin is a proxy for phytoplankton, which is a really big component of these algal blooms that we see. So we had some pretty interesting results when we're just looking at chlorophyll A. We see there's a pretty clear distinction. We put that dotted line in there just to show that separation, where we had some treatment groups that had a lot more growth than other treatment groups. And what's interesting about the division is the groups that had a lot more growth were all the groups that received additional phosphorus as part of the treatment. And all the groups that were below that dotted line, so they didn't have a lot of growth, those are the groups that didn't have any phosphorus added to them. So already we kind of see that there is a phosphorus limitation, specifically for cyanobacterial growth. We see a pretty similar distinction with phycocyanin, though it's not as strong as with chlorophyll A. So we looked at this distinction. We looked at the amount of phosphorus and nitrogen that was in each of these samples. Specifically, we're talking about soluble reactive phosphorus and dissolved inorganic nitrogen. Those just mean the bioavailable forms of these nutrients, and this is what we'll keep referring to them as in the future. So we looked at this distinction, looked at the amount of phosphorus and nitrogen in the groups, and we set these thresholds where we know if there's below or if there's about this much phosphorus, we're not going to see a lot of growth. And if there's more, we are going to see growth. So we took these thresholds and we looked at some of the weekly data that we had from another project in our lab. And we can see specifically with phosphorus in this top graph, that especially during summer and all the way into fall, we're far exceeding this threshold that we've set. And this is also when we're seeing these really intense algal blooms. This is the worst time of year for them, usually. So we see when there's a lot more phosphorus in the water, we're seeing these really intense algal blooms. We're not as worried about nitrogen because we're around the threshold that we've set for most of the year, except for in spring and a little bit into early summer. So with that part of our experiment, we found that phosphorus is a really limiting nutrient for algal growth, but we also set dilution bioassay as a viable method to test for these thresholds. One of the other questions that we wanted to address, it was about the toxins that are produced by these algal blooms. And we wanted to figure out why these have higher concentrations when they do. So what's encouraging the production of these toxins? And what was interesting is out of all the parameters that we measured, we couldn't find any significant correlation with the amount of toxins that were in our samples. Even though we know that cyanobacteria produce these toxins, there was no correlation between overall biomass, cyanobacterial abundance, or any other parameters that we measured. So it's still a bit of a mystery to us. In looking at these graphs, though, we can almost see that there's a bit of a seasonal aspect to when these toxins are produced. But it's interesting because if we look specifically at late summer, microcystin concentrations have a big dip in that time of year, but antitoxin A concentrations have a huge peak. So we're seeing these trends, but we still haven't been able to attribute them to anything specific. So that was just part of the experiments that we did and part of the data that we collected. But we did establish that phosphorus was the limiting nutrient for cyanobacterial growth. So we believe that if we reduce the amount of phosphorus that we're putting into the lake, that we can significantly reduce the amount of blooms that are occurring and the intensity of these blooms. But if we look at this threshold that we set and we compare that to the current state guidelines for the wastewater treatment plants around Utah Lake, there's an order of magnitude difference. And if we were to tell these wastewater treatment plants that they need to reduce the amount of phosphorus that they're letting flow into the lake, they would have to reduce their output by about 90%. So it is a big jump, but we believe that if we reduce the amount of phosphorus that we put into the lake to this threshold, that we won't see these strong algal blooms. And now there's more research that needs to be done, right? We didn't set the determinants for toxin production, so we still don't know exactly why those occur when they do and at what concentrations. But we did set dilution bioassay as a viable way to test for the thresholds for algal blooms. So we believe that more of the experiments need to be done so we can further pinpoint exactly where these thresholds are for algal growth in Utah Lake. And in doing so, we can be more exact in our management practices and we can really get these algal blooms in Utah Lake under control. So thank you for listening to my presentation. Feel free to check out the rest of my poster. Thanks.